Good morning everyone. Today I am presenting slide number 8. History of that patient was 20-year-old male presented with pain in abdomen and fever. The slide given to us was of peripheral smear showing normocytic hypochromic RBCs with normal platelet count with leukocytopenia. On high power view, we can clearly see ring forms in RBCs. In this image, we can clearly see ring forms having chromatid dots along with it, hence the diagnosis is malarial parasite. Introduction. Malaria is endemic in tropical and subtropical developing countries where it is a major public health problem. Every year, there are about 300 to 500 million clinical cases of malaria with 1 to 2 million deaths, most of them in Africa. There are 4 species of malaria that infect humans: Plasmodium vivax, Plasmodium falciparum, Plasmodium ovale, and Plasmodium malariae. The most common prevalent form in India is Plasmodium vivax. Life cycle of malarial parasite. Life history of malarial parasite consists of 2 cycles of development, asexual cycle and sexual cycle. Asexual cycle occurs in liver cells and RBCs of infected human. Therefore, humans are intermediate host of malarial parasite. There are 4 stages of human cycle: pre-erythrocytic schizogony, erythrocytic schizogony, gametogony, and exoerythrocytic schizogony. In pre-erythrocytic schizogony, inoculated sporozoites rapidly leave the circulation to enter liver cells where they develop into hepatic schizont. Hepatic schizont rupture to release numerous merozoites in the circulation. Merozoites released from rupture of hepatic schizont enter the red blood cells via specific surface receptors. These merozoites become tropozoites that utilize red cell contents for their metabolism. After several cycles of erythrocytic schizogony, some merozoites, instead of developing into tropozoites, transform into male and female gametes, and this is called as gametogony. In Plasmodium vivax and Plasmodium ovale, some of the sporozoites in the liver cells persist dormant. These dormant forms of liver cells is called as hypnozoite. They become active and develop into schizont a few days later, and this schizont rupture releases merozoites and cause relapse. This is the another image showing life cycle of the malarial parasite. Clinical features. Clinically, malaria is characterized by paroxysms of high-grade fever along with anemia and splenomegaly. Malaria is the most common cause of splenomegaly in the developing countries. There are 3 stages of typical malarial paroxysms: cold stage, hot stage, and sweating stage. In cold stage, there is sudden onset of fever, shivering, and extreme cold. In hot stage, fever rises to maximum, and in sweating stage, patient perspires profusely and temperature falls. Mode of transmission. Malaria is transmitted by bite of infected female Anopheles mosquitoes. There are various methods of diagnosis. Some of them are microscopy, rapid diagnostic test, and nucleic acid amplification test. Microscopy. It is the gold standard for diagnosis of malaria. Blood smear should be obtained before the next expected febrile paroxysm or at the onset of fever and chills. One thick smear for detection of parasite and one thin smear for definitive identification of species should be prepared. Thick smear is more sensitive than thin smear for detection of malarial parasite, particularly if they are few in number. This is the image showing thick and thin blood smear. Parasite density. In thick, in thick smear, number of parasites is counted till 200 white blood cells are observed. Number of parasites present per microliter of blood is equal to total leukocyte count per microliter into number of parasites divided by 200. Usually, total leukocyte count is taken as 8,000 per microliter and number of parasites is multiplied by 40 to get results. In thin smear, number of parasites among 1000 red cells is counted and reported as percentage. This is the image showing various morphologies of Plasmodium species. In this image, we can see the ring forms, the tropocyte forms, schizones and gametocytes of various species. Rapid diagnostic test. These tests detect antigens of malarial parasite by immunochromatographical method. The antigens against which commercial test kits are currently available are histidine-rich protein 2 synthesized by asexual blood stages and young gametocytes of Plasmodium falciparum, parasite lactate dehydrogenase and parasite altolase. This is the image showing rapid diagnostic test. Other diagnostic tests are detection of nucleic acid sequences of malarial parasites, serological method, and flow cytometry. Thank you.